Hello everyone. We, Collaborations Pharmaceuticals, are developing an enzyme replacement therapy for CLN1 Batten disease. CLN1 Batten is an ultra-rare neurodegenerative disorder characterized by progressive intellectual and motor deterioration, seizures, loss of vision, and early death. It is caused due to mutation in the CLN1 gene, which codes for the lysosomal enzyme palmitoyl protein thioesterase 1 or PPT1, resulting in a reduction or absence of enzyme activity in CLN1 patients. CLN1 presents in humans as early as 6 and 24 months of age. There is no treatment available for CLN1 Batten patients so far. We are performing IND-enabling toxicology studies using recombinant PPT1 enzyme and are working towards phase 1 clinical trials. Methods: PPT1 protein was generated using a CHO-ZN cell line stably transfected with plasmid expressing recombinant human PPT1. The protein was purified by hydrophobic charge induction chromatography, cation exchange, and hydrophobic interaction chromatography. Analytical size exclusion chromatography was used to determine purity of the protein. Activity of recombinant PPT1 was determined using an enzyme assay in a 30-minute reaction to determine specific activity of the recombinant enzyme. Cellular uptake of recombinant PPT1 was determined by treating cells in the presence or absence of mannose-6-phosphate. Enzyme activity was measured in cell lysates for 60 minutes. Specific activity was reported as units of substrate converted per meg of lysate and used to calculate uptake of recombinant PPT1. Results: Figure 1 shows an overview of the timeline and progress for recombinant PPT1 enzyme replacement therapy. We have completed preclinical efficacy studies in animal models of CLN1 Batten, as well as process development, analytical development, and formulation of recombinant PPT1 for large-scale production. We have produced GLP quality recombinant PPT1 protein and have characterized it extensively to enable IND-enabling toxicology studies in non-human primates, which is ongoing. GMP production of protein for clinical trials is underway. Figure 2 shows analytical characterization and stability for recombinant PPT1. Recombinant PPT1 appears as a clear solution and is stable at pH 5.5 to 7.5 at temperatures 5 degrees Celsius and minus 70 degrees Celsius for at least a year. Recombinant PPT1 is formulated in artificial CSF in which it maintains high purity, stability, and activity. Figure 3 shows cellular uptake of recombinant PPT1 in cell lines from different species. We have demonstrated that uptake of recombinant PPT1 is dependent on the mannose-6-phosphate pathway in human wild-type neurons, PPT1 deficient neurons, and monkey astrocytes as measured by enzyme activity and key uptake values. Table 1 shows the tests performed to assess purity, potency, quality, and content of GLP quality recombinant PPT1 produced for toxicology studies. Conclusion: We have completed GLP-grade manufacturing of recombinant PPT1 and are currently performing analytical validation and quality control for future GMP production. Cellular uptake of recombinant PPT1 is dependent on the mannose-6-phosphate receptor in primary human neurons, CLN1 null fibroblasts, as well as monkey astrocytes as measured by enzyme activity in these cells. We are performing GLP toxicology studies in non-human primates and then will undertake GMP manufacturing for a future clinical trial. I acknowledge all the people and organizations involved in this endeavor, especially STC Biologics, for recombinant PPT1 large-scale production and development. I thank our CEO, Sean Eakins, all the patient families for their support, and the NIH for funding. You can get in touch with me at renuka@collaborationspharma.com if you have any questions. Please visit the live poster session on Wednesday, February 5th between 3:30 and 5:30 p.m. Pacific Standard Time at the Manchester Grand Hyatt Hotel at San Diego. We have also developed a patient registry to recruit patients. Please visit poster number 325 for details on patient registry and how to enroll. Thank you. Bye-bye.